Hi, everybody. Welcome. This is the SBGrid weekly webinar series. Hope everybody is staying safe and managing all the things that are going on in the world outside of science reasonably well. So today we have Dhirendra Simanshu from the Ras Structural Biology Group Lead from National Cancer Institute and from the Ras Initiative. So thank you for joining us and for agreeing to tell us about your science. And with that, I will turn it over to you.
Thanks, Pete. Let me see if you all can see me.
Yep, screen share looks good again.
Okay. Hello, everyone. My name is Dhirendra Simanshu, and I am at NCI Ras Initiative, Frederick National Laboratory for Cancer Research at Frederick, Maryland. And let me start by thanking Pete for the introduction and myself for the invitation to share some of our work in this forum. I've been using SBGrid for more than 10 years, initially as a part of my postdoctoral work at Memorial Sloan Kettering, and then since 2014 here within my team, SBGrid has been very useful for me, especially it allows me to really focus more on science and less on time on trying to make sure that software compiles well and they're updated regularly. So from that perspective, it always was no brainer for me to use it when I started my own lab here at Frederick, Maryland. So thanks to the team for doing what they do. It really saves a lot of time for many of us. So today I'll talk about some of the work which we've been doing here at Ras Initiative, mostly from the structural and biology perspective. Just to give you some idea of what the initiative is. So Ras Initiative is a national mission program at Frederick National Lab. As many of you know that Ras proteins are mostly comes in three flavors, K-Ras, H-Ras, and N-Ras. And they play an important role in cell signaling. Ras happens to be the most common oncogene in human cancers, responsible for almost 20 to 30% of all human cancers. And the Ras mutations are very common in pancreatic, colorectal, and lung cancer. Ras has been the subject of intensive studies for more than 35 years, but despite many of these efforts, not just to understand Ras biology, but also to understand and explore ways to target Ras-driven cancer, there hasn't been significant improvement till recently in drugging Ras-driven cancers. Around 2013, Harald Wormes initiated the idea of establishing a national mission program at Frederick National Lab. And since many of the biotech and pharma were giving up at that time on Ras driven cancer, they decided to start a national mission program here to find novel ways to attack Ras driven cancer. And this is what the situation has been unfortunately in this case, that anytime you have a mutant Ras, there is not much we have got for them. And till recently, at least now for a G12C mutant allele, now we have got something in the clinical trial, and hopefully that will come out soon. And hopefully that will also give ways to at least ideas to target other mutant alleles of Ras. Just to give you where things are in context of Ras, as among the three Ras proteins which has been involved in Ras-driven cancers, the K-Ras is the prominent one. It is responsible for 86% of all human cancers. N-Ras is the second one, 11%, and H-Ras is 3%. Whereas the structural work, at least till 2014, was very skewed towards H-Ras. Most of the structural work were done on H-Ras, were very little on K-Ras, and very, very little on N-Ras, maybe just a single structure of N-Ras. And it is mainly because due to the reason that H-Ras is much easier to express, purify, and crystallize. So maybe that's the reason. And there is a lot of structural information you can extrapolate from each other. So it's not that very different, but there are a few differences, and that at times is very important for the Ras function as well. So our very conscious effort has been to focus all our efforts on K-Ras and not from the biology perspective, but also from drug discovery perspective. For those of you who are not very familiar with Ras biology, once the RTK gets activated and then it activates to the GAP protein, the guanine exchange factor, which is mostly controlled by SOS. SOS loads the Ras from GDP to GTP and makes it active. The green one becomes active, and then it signals, sorry, downstream via RAF, PI3K mainly, and Ras-GDS. There are many effectors of Ras which has been known. There are more than 10, 12 effectors. And the Ras cycles between active and inactive form via SOS and via GAP proteins. And the two Ras gaps which have been very well studied are, one is neurofibromine, in abbreviation we call it NF1, or Ras-A1, which is also known as P120 gap. So I will talk mostly about NF1 neurofibromine today, and I'll also talk about Ras-Raf interaction. And when we started at that time, at 2014, there wasn't really a lot of structural data on K-Ras mutant in the active state. There were more in the inactive state, and there were HRAS structure, but not the K-Ras structure. And then there was not a single structure of any Ras protein where you can actually see the hypervariable region, the last 20 amino acids, and the farnesylation and the carboxymethylations, which is occurred at the C-terminal end of it. And most of, as I said, most of the structural work were on HRAS, so there was not really much structural work, or in fact any, on wild type K-Ras or oncogenic mutant K-Ras with effectors or regulators. And again, till recently, there was not really much structural information on full length RAF, either free or in complex with RAS. And then RAF remains the major target for RAF driven cancer, Ras-Raf interaction, how RAS activates RAF kinase. So there was not really much structural insights till recently, when a lot of cryo-EM studies provided significant insights on how RAS activates RAF kinase. And all these have changed significantly in the last 5 to 6 years. In fact, just to tell you that K-Ras structure, which was 31 in case of 2014, has reached 200 now, and this is still around 180. So just in the last 6 years, the structural work on K-Ras has gone almost 7-8 times and deposited in the protein beta data bank. And I expect this to really exponentially grow in the next 5 to 10 years when we actually get much more drug discovery work done on K-Ras. So starting with one of our major contribution in the beginning was solving the first full-length structure of K-Ras. And this is a schematic on the left side, which I'm showing you that this is the G domain, the globular GTPase domain of Ras. And then it is followed by approximately 20 amino acid long hypervariable region. And this region differs between different Ras isoforms. And that actually gives them some sort of a unique biochemical and cellular properties. And many of these Ras isoforms, at least in case of K-Ras, K-Ras also has two splice isoforms. One is K-Ras 4B and one is K-Ras 4A. The K-Ras 4B is the one which is actually responsible for most of the human cancers. And as you can see, it has a polylysine stretch in this region, which plays an important role in membrane interaction. And also there is a farnesylation at site at the terminal cysteine, and then the terminal cysteine also gets carboxymethylated. And these modifications are important for membrane anchoring of Ras, and all Ras biology happens at membrane. So in some aspect, this region plays an important role in Ras biology. This is the structure of first full-length, fully modified Ras in complex with PD delta. PD delta is a chaperone protein which actually takes K-Ras, fully modified full-length K-Ras from ER to the membrane for membrane anchoring, because some of these modifications happens at ER. And as you can see that when we solve the structure, we actually, people had believed all along that this region is highly disordered structure and doesn't really form any secondary structure. But we did see that at least in one crystal form, that most of these residues up to 179 could form an extended helix, alpha 5 helix, and the last 6 to 7 residue could actually form a loop. And in this case, the protein gets solubilized in the aqueous environment of cytoplasm by sealing this lipid tail in the core of the PD delta protein. And as I said, PD delta stands for phosphodiesterase delta subunit, and it binds and solubilizes purine-related proteins. And this is not just unique to Ras, but it does actually almost 2% of the proteome, and then felicitates its transfer or diffusion in the cytoplasm. So this was one of the work which actually allows us to see K-Ras in the first time in the head-to-tail form. What I'm going to present you today, some of our work which we have published in just last 12 months. One of them is related to GAP. This is neurofibromine, and the second one is on RAF kinase. So related to GAP plays a most important role actually in RAS cycle, because it actually converts an active RAS to inactive RAS. And only when the RAS is unable to make GTP to GDP conversion, it actually becomes oncogenic. So for most of the RAS-driven cancer, the bigger loss of function is in its ability to undergo GAP-mediated nucleotide cleavage, converting from GTP to GDP. And there was a protein which was discovered in 2001 called SBGrid. It stands for a sprout-related domain, a sprout-related protein which contains EVH1 domain. And it was shown that it actually acts as a negative regulator of Ras-MAPK pathway. And later on, it was shown it actually works via NF1. And then recently, we actually solved a structure of a ternary complex of Ras, NF1, and SBGrid, and showed how actually this complex works at the membrane. Which I'll present. In the second part, I'll show a structural work of Ras in complex with RAF. RAF interacts to Ras via two domains, Ras binding domain and cysteine-rich domain. So we use that two tandem domains to solve the structure. This is RBD, Ras binding domain, and cysteine-rich domain. So that I'll discuss in the end. So starting with the first part of the work. So this is the model of which was proposed finally in 2012. So as I said, the protein was discovered in 2001 in a landmark paper where it was actually shown that SBGrid is a negative regulator of Ras-MAPK pathway. But for 10 years, it wasn't very clear how actually it acts as a negative regulator. There were theories based on it directly binds to RAF, it could, and then it controls the MAPK pathway. Then some people proposed GAP, and then finally in 2012, Frank McCormick's lab at UCSF showed that that SBGrid, which has three domains called EVH1 domain, a KIT binding domain, and a sprouty domain. So the sprouty domain binds to the membrane, whereas EVH1 domain actually binds to the neurofibromine NF1, and then possibly brings it to the membrane, where actually NF1 hydrolyzes GTP to GDP. And that's how the EVH1 or SBGrid controls the Ras-MAPK pathway. And it was also shown that germline loss of function mutation in SBGrid causes a very similar disease, which has been shown to occur because of mutation in neurofibromine. That disease is called neurofibromatosis type 1. So just to give you some glimpse of what these two diseases. So, many of you have heard about Ras-driven cancer, but there is a separate set of collection of diseases called rasopathy, which happens, in germline mutations in various proteins, which actually affects Ras-MAPK signaling. And this is a schematic which actually shows those genes. As you can see, the NF1 causes neurofibromatosis type 1 kind of disease. And this is the SBGrid, which counts a disease which was discovered by Ligius, and on his name, it is called Ligius syndrome. And many of these mutations are in Ligius syndrome occur in the SBGrid protein. And we'll talk about some of them in the later slide. And similarly, there are other rasopathy syndrome, which will not go today, like Leopard, Noonan, Costello, which is again caused by different genes. So coming back to how does SBGrid really acts as a negative regulator. And this is a paper published by Klaus Efsack and Frank McCormick in 2016, where they showed that what are the reasons responsible for NF1 SBGrid interaction. So let me start by showing you the domain organization of these two proteins. So neurofibromine or NF1 is a very long protein. It's almost 3,000 long protein. And in the middle of it, there is a small domain called GRD, gap related domain. And this gap related domain again has a central gap domain, which actually has the gap activity. And then on both sides, there are extra amino acids, which is actually just present. And so they took this whole GRD, and which you can see that this is GRD minimum, which is the central catalytic part of it. And then you can actually, in one case, they removed the C-terminus extra part. In one case, they removed N-terminus extra part. And in one case, they removed both of them. And then they do IP. And what they found that the minimum part, which is GRD, the bottom one doesn't have the ability to interact with the spread one. The only one which actually interacts with the spread one is the one which has both N extra part and C extra part. And based on that, they basically proposed this model that the central part, which is in this blue, dark blue, is actually the one which interacts to Ras, has the gap activity. And then the N and C terminal part fold together at one end of it. And that's where the EVH1 domain of a spread one binds to it. And this is the EVH1 domain. So based on this work, we made multiple constructs of NF1, a spread one, and then try to form a stable ternary complex. So we just not went with the binary complex, but we went with the ternary complex and tried to capture all three of them. Finally, we succeeded with this combination, where we actually tried to use a spread one EVH1 domain. We use K-Ras active form and GMP PNP, because in that case, it can actually interact with NF1, forms a stable complex. And then we used a GRD, the gap related domain, which is around 330 amino acid in between here, somewhere from 1200 to 1530. 1203 to 1530, that's the construct actually we used it for crystallization. And then eventually with that, we actually could express and purify a very stable complex of all three proteins and crystallize and solve the structure. And this is the structure of that ternary complex, where you see the K-Ras is shown in yellow. The green part is NF1, neurofibromine, GRD. And then this is the pink part is the spread one EVH1 domain. Overall, a structure is basically similar to what were proposed based on those mutagenesis studies. As you can see, the light green part is the one which forms the catalytic part, which forms a stable complex with Ras. This N extra domain and the C extra domain, both of them come together and actually form a small globular domain at one end of it. And this is called extra domain here. And that interacts with spread one. What we did see that, which was not observed earlier, was that there was a part of catalytic domain as well, which actually interacts with the spread one, not just the extra domain. Both extra domain and the central part interacts with it. But the interaction formed with the extra domain is important for the complex formation. So it looks like if you remove either NX or CX, you will lose the binding between a spread one and NF1. We went ahead and analyzed the protein-protein interaction. As many of you who knows about Ras, there are two switch regions in Ras. One is called switch 1, which is somewhere between 28 to 36 or 30 to 40, somewhere around. And then switch 2, which is around 60 to 76. And these two are the most critical regions which are often involved in interaction with the regulatory proteins like GAP and GAF, as well as effector proteins like RAF and PI3K. So when we look at some of those interactions, as you can see that switch 1 and switch 2 region both are involved in Ras NF1 interaction, but majority of the interaction is via switch 1. As you can see, there are multiple interactions here, and these are the switch 2 interactions which are formed here. The binding affinity between Ras and NF1 is 1 micromolar, so it's a significantly stable interaction. We went ahead and tried to understand what are the most critical interactions between at Ras and F1 interface. as I pointed out, that both switch 1 and switch 2, we mutated few residues from both ends, from switch 1 side and switch 2 side, and actually observed that a mutation of D38A, Y40A from the switch 1 region, almost significant loss of binding, and similarly from switch to E63A and Y64A results in significant loss of binding. So it actually shows that that both switch 1 and switch 2 are critical for Ras-GAP interaction, and any perturbation at any of these regions could potentially disrupt the Ras-GAP complex. Similarly, we also looked at some of the interface residues from NF1 side. So what you're looking at it here is that you're looking at Ras in electrostatic surface representation, and you're looking now NF1 as a cartoon representation or ribbon representation. And one of the critical residue in NF1 is R1276. This is the catalytic residue which actually attacks the gamma phosphate for the nucleophilic reaction. So as you can see, that mutation of this, although loses the catalytic activity, but it doesn't really affects the protein-protein interaction. Still, you can see 1.8, 4.46. So this is wild type, this is mutation, just maybe twofold decrease. But then there are other residues on NF1, especially which binds to the switch 1 and switch 2 region of it, and especially these three residues, which are shown here, 1436 to alanine. This is a lysine to a glutamate mutation, a single mutation, complete loss of binding. As you can see that this is actually, this lysine enters from the top of the nucleotide binding pocket. There is a small hole there, and which actually tries to enter. And if you just make a charge reversal mutation, this results in complete loss of binding. This is around switch. This is around switch 1, and this is somewhere around switch 2 region. So again, from NF1 side as well, both regions are important for binding. Next, we went to start look at what happens on the NF1 spread 1 interface, which is completely different from this. And our all binding data, which I have no time to present, but we do have data where we saw that the binding of binary complex or the formation of a binary complex has no effect on the third partner. So you can actually form a NF1 K-Ras complex and it still binds to spread 1 with the same affinity, or you can form a spread 1 NF1 complex and it still binds to K-Ras with a similar affinity. So in this case, again, in fact, the binding affinity of NF1 spread 1 is even is in the much lower. It's somewhere around 290 nanomolar. So that interaction interface is still quite extensive and much more actually higher affinity than K-Ras GRD interface. As I mentioned, that mutation in a spread 1 causes Legius syndrome, and these are the mutations which have been mapped in case of Legius syndrome patients. And because in our structural data, we only have EVH1 domain, I'm just looking at the mutation in this region. And many of these residues, which is marked here, are actually at the protein-protein interface. And we made some of the similar mutation like this R24Q, G30R, which is somewhere here, then T102R, G100D, and we made some of these mutations and checked the binding affinity. And in many cases, the binding, in this case, it was around 10-fold, in this case, 100-fold. T102R resulted in complete loss of binding. And in 2 cases, 2 other mutations which have been seen in case of Legius syndrome patients, W31C and G100D, these 2 proteins, in fact, we couldn't even make those proteins. So it looks like these mutations completely destabilize the EVH1 domain and potentially protein gets degraded. So some of these interface residues actually explained rationale for why some of these mutations in Legius syndrome could perturb spread 1 NF1 interaction. So as I mentioned that there are 2 very well-studied Ras gaps. One is NF1 and the second one is Ras-A1, also known as T120 gap. And what has been shown over the years that spread 1 interacts to NF1, but it doesn't interact to Ras-A1. And we haven't understood why. So when you look at the structure sequence alignment of these 2 proteins, as you can see that they actually share significant significant similarity in the core of the protein where the catalytic part is. But at both ends, NX and CX, which I have marked by red, they are not really that conserved. In fact, very few, 3 or 4 residue on the N and 2 residue on the C side is actually just totally conserved. And this is the structural superposition. As you can see that the core part does look quite well aligned, but this is the extra domain which actually interacts with the spread one. And as you can see that if I zoom this extra domain in a separate picture, you can image, you can see that these helices move significantly with respect to each other. And also some of the key residue which interacts here, which is again highlighted here, some of these are different in case of P120 gap. And some of these structural changes are likely to be responsible for spread one having a specificity towards NF1, but not towards Ras-A1 or P120 gap. While we were working on in this, Frank McCormick's lab at UCSF has a very interesting results also. They have been looking at the effect of oncogenic EGFR on the spread one NF1 interaction. They had some preliminary data which suggested that oncogenic EGFR could potentially disrupt NF1 spread one interaction. So when they actually did this IP, when they expressed NF1 alone and then NF1 along with EGFR L858R mutation, they couldn't really see NF1 spread one coming in. So it was likely that a spread one was being modified by EGFR somehow in case of oncogenic EGFR. So they went ahead and did PTM phosphorylation analysis, and here they actually expressed the spread one flag tag with GFP, and then did oncogenic EGFR, did a flag IP, and then did LCMS-MS. And based on that, they actually identified 5 different phosphorylation sites on a full length NF1. Among these, at least this one, S105, was located on the EVH1 domain and was part of the ternary complex which we had solved. So then we went ahead and looked at this particular serine in case of a different organism, and it does look like that it is actually conserved across the spread one from fish to human. And then we went ahead and made a phosphomimetic mutation and converted a spread one to an aspartate or glutamate, or also made a phosphodeficient mutation of S105A. And as you can see in this data that a wild type spread one binds, the phosphodeficient binds, but as you mutate that into a phosphomimetic S10D or S10E, it does not bind to the neurofibromine. And when we map this particular residue on our ternary complex structure, it actually points, this particular serine points towards a very acidic patch on the NF1 side. So any mutation of this serine to an aspartate or glutamate likely disrupt this interaction. So based on this study, this is our current model, that when you have an oncogenic EGFR, it releases it through a cascade of pathways. It somehow, we don't really know what's the intermediary partner, the components are, but it probably phosphorylates a spread one, at least an EVH1 domain at S105. And this particular phosphorylation site prevents NF1 spread one interaction. And also we showed that a spread one by EVH1 domain brings NF1 to the membrane, and then it can convert Ras-GTP to GDP. So one of the roles of a spread one is to really bring NF1 to the membrane and keep it close to the, maybe near to the Ras to regulate the Ras activation cycle. There is some effort going on in Frank's lab here, is trying to identify some of the proteins which could be involved in the phosphorylation of a spread one. But that was the part one of the study. I'll quickly move into the second part, where we have now focused into the second component of Ras pathway is downstream signaling. And among downstream signaling, as many of you know, a majority of the signaling events happen via this first one, Ras-Raf pathway. And there has been a lot of work. I didn't have time to review this, but in a couple of years, there was a significant structural work published by various groups, Michael X group, John Kurian's group, Genentech's group, with some of the cryo crystallography work on full-length BRAF in both active state and inactive state. One of the challenge in that some of those studies that even though they used a full-length RAF, but in the cryo map, most of the time the first half of RAF was not very well visible, except in one of the studies from Michael X group, the CRD was as visible. But in all other works, they couldn't really see the first half of the RAF. And since none of these studies included RAS, we still did not fully understand how does Ras-Raf interaction takes place. So to fill some of these knowledge gaps, we decided to initiate a structural work on this particular conserved region of RAF, which is called CR1. This is called CR2, and the kinase domain is called CR3. So we took the CR1, which is actually the RAS interacting site. RBD has been well characterized as a RAS binding domain, and whereas the cysteine-rich domain has been shown to play a major role in membrane anchoring. And this is what the RAS switch 1, switch 2, and then HVR. One thing, but we saw that RAF RBD, at least in this particular condition, and SPR binding assays, we saw around 370 nanomolar affinity with just the RAS binding domain. And when you include the cysteine-rich domain as well, we did see a twofold increase in the binding affinity in case of the two tandem domains of RAS binding domain and cysteine-rich domain. And as you can see that if you compare, the binding response does increase when you have CRD. The question is that there is the role for CRD has been mainly to propose to actually act as a membrane anchoring domain. So it wasn't very clear when we had this data, is the CRD is really stabilizing RBD and then resulting in higher affinity, or is CRD is also contributing to the Ras-Raf interaction interface. So when we saw the structure, it actually came as quite a bit surprise to us that not just the RAS binding domain, which has been all along shown to be a major contributor of Ras-Raf interaction, but it is the CRD also which actually contributes significantly to Ras-Raf interaction interface. So if you look at, this is the ribbon representation and this is the surface representation, and as you can see that the extent of RAS RBD interaction, which is cyan, and this is the switch 1, which is in the magenta, and you can see the green is CRD and then RAS. It does look very similar in the surface area wise, but number of hydrogen bonds and the salt bridges are actually a little more on the RBD side. So RBD still plays a major role, but it does look like that the CRD does play a supportive role in increasing the affinity of Ras-Raf interaction. And we were a little worried that, is this some kind of an artifact? So we actually saw the structure in two different crystal form, and when we superpose them, they actually superpose almost on top of each other. So this interaction interface was not really some sort of an artifact of crystallography. We went ahead and tried to compare the RAS RBD interaction interface, which has been actually shown before in context of HRAS and isolated RBD domain. And many of those interactions remains, same. We actually also, these are K-Ras, which is formed again by many of these switch 1 residues, E31, D33, E37, and then some of them after switch 1 is like S39 and R41. And as you can see that the 2 beta strand, which has been a very central theme in RAS effector interaction, that one of the beta strands of RAS and one of the beta strands of effector, they basically come and line each other and they form a extended beta seat. And that's how there's a lot of main chain interaction involved in that as well. And among all RAS effector interactions, the Ras-Raf interaction is the strongest, and it goes somewhere around, depending on which method you use, you can see almost 100 nanomolar to 300 nanomolar. No other RAS effector interactions, in fact, is this strong. Most of them goes into much weaker, 1 micromolar, 2 micromolar, 10 micromolar. So in some way, Ras-Raf interaction is very strong. So mutation of some of these residues in the RAF RBD-CRD tandem domain also showed a very similar result what we had seen before, that if you just make a single mutation in RBD R89L, it completely dissociates RBD-CRD interaction. So CRD is still not strong enough to hold that tandem domain to RAS. Just RBD is still plays a major role. And these 3 additional mutations resulted in significant loss of activity. So this is what you're looking at, 300 micromolar, and as you can see, this is almost 1 micromolar, this is around 500 micromolar, this is 2 micromolar, this is complete loss of binding. And if you look at these RBD, again, many of you probably know that RAF has 3 flavors again, A-RAF, B-RAF, and C-RAF. What we did work on C-RAF, also known as RAF1, but there are B-RAF and A-RAF, and many of these residues which interacts with RAS, which I have shown in a RBD K-Ras in the triangle overlays with the RBD CRD and then with the linker. But as you can see, many of these residues are conserved across 3 RAF isoforms. So most likely the interaction interface between RAS and RAF remains same in all 3 RAF isoforms. I think the most interesting part on this work was focused on CRD. The only structure of CRD, RAF CRD, was solved more than 20 years ago by NMR. And we used that particular structure as a starting model to phase the CRD part. And as you can see, the overall structural superposition with the NMR structure is quite well. Most of the secondary structure fit quite well. Zinc sites are quite close to each other. Some of the loops do take quite different path, but maybe some of them because of they are part of RBD, and whereas the NMR work was done in an isolated CRD. And as expected, we saw 2 zinc atoms, again, forming by 3cysteine and a histidine here, one located at the N terminal, the N and C terminal junction here, and one of them is near this helical part, which is stabilized here. And as you can see that what came again as a surprise, that so far RAS effector interactions have been either limited to switch 1, and in some case 1 or 2 interactions to the switch 2. But what happens, in this case, that a lot of RAS CRD interaction is not really part of switch 1 or switch 2. RAS CRD interactions are mostly found by regions between switch 1 and switch 2, what we call it inter-switch region. And then on the last helix called alpha 5, which is the 5th helix and the last helix of this. And as you can see, this is a RAS protein, and these are the residue which interacts to CRD. And as you can see, these are located 41 to 48 here. And then some of them before switch 1, which is 24, 23, 26. And then the last helical part is 149, D153, Y147. So in that aspect, Ras-Raf interaction, at least Ras-CRD, from what we have seen in case of other RAS effector complexes. And again, from the CRD side, the residue which interacts to RAS, again shown in the pink on the green background. And these are again spread all across, not just limited to one part of the or one stretch of the amino acid. So it's quite a spread all around. Just to give you a perspective that that RAS CRD interaction still contains 8 hydrogen bonds. And although the isolated CRD still doesn't bind to K-Ras with high affinity like RBD, but it is not all hydrophobic. There are at least 8 hydrogen bonds that we saw, and there is significant hydrophobic interactions as well. What is so interesting about CRD is that this is just a 50 amino acid long domain, and it interacts with a large number of proteins. When the RAF is inactive state, like B-RAF, as was shown in Michael X's work, they showed that how CRD is actually trapped in the B-RAF between B-RAF and 1433. And they actually showed that there is a lot of residue which actually forms that contact. We actually mapped those interactions from CRD 1433 interactions, which are shown in squares. And then in hexagon, we have mapped CRD B-RAF kinase domain, and you can see those are in hexagons. And then we went ahead and mapped what we saw in these crystal structures like CRD RAS contact, then CRD RBD contact. We did see some interaction between CRD and RBD as well. That is one part which we again were a little surprised initially with what we thought or what was believed, that these two domains are actually two bits on a string. They're not really close to each other. But what we saw, at least in the surface representation, that actually they do seems to form more like an extended structure. And these two domains are actually quite close to each other, not really a two bits on a string. And in that case, as you can see that, and then there is a small linker between these two, which actually brings again both domains close to each other. And as you can see that all Ras CRD interaction, probably except this one particular residue, this arginine, which overlaps with CRD 1433 contact, none of the Ras CRD residue, which is again on the C terminal side and a few on the N terminal side, actually overlaps with the CRD ability to bind to either 1433 or CRD ability to bind to kinase. I'll come back to this on a later slide, but it shows that CRD, Ras could bind to CRD even when CRD is potentially part of the inactive complex when it is interacting with the 1433 and kinase domain, because there is not a lot of overlap between binding sites. Again, considering that Ras CRD interface was completely it has not been studied before, we went ahead and again made some mutation on both Ras side and on the CRD side. And as you can see that even though we made certain mutations here, but since RBD contribution is still very strong, the effect of these CRD mutations were very little. At least here, you can see these are CRD mutations starting from F141 to these. And as you can see that at least two of them, K179 and F141, had a 3 to 4-fold lower binding, but others didn't have really significant reduction. It's also mainly because RBD still is holding those two proteins quite strongly. So even if CRD comes off, it probably does not dissociate completely. It still has a significant interaction. And similarly, we actually chose residues on Ras, which actually forms Ras CRD interaction, like the residue present in interswitch region, this K42, Q43, V45, and then two of them on the alpha 5. And as you can see, at least these two residues, one on interswitch V45E and D153A on alpha 5 did reduce the binding affinity to 2 to 3-fold. So it does look like that Ras CRD interaction does at least diminishes the Ras-Raf interaction, these mutations. One of the work which we wanted to do, and it was inspired from some of those work which was published 20 to 30 years ago, what is the role of Ras CRD interaction on Ras mediated RAF activation? And there were some reports that if you perturb Ras CRD interaction, it could actually potentially have some effect on RAF activation. So we decided to take a study this based on structural data. So based on the protein interaction interface, we made a similar mutation our SPR binding studies data. And these we tested what is the effect of these RBD mutations on RAF activation and what is the effect of these CRD mutation on RAF activation. And similarly, we also made some of those on the K-Ras side and mostly focused on K-Ras CRD interaction. And what we saw that at least in case of here, if you see here, this is the wild type, this is 100% full kinase activity. And as you can see that in case of RBD, of course, some of these mutations which perturbed Ras RBD interaction has reduced RAF activity. But we also saw that similarly T178A and maybe even this L136A, which is in the linker region, which is actually does not perturb Ras RBD interaction. So the affinity between Ras and RAF in these two mutants remains same, but still they have almost as reduced activity as you can see with even N64A, which reduces Ras RBD interaction almost by 10-fold. So it does look like that Ras CRD interaction still plays an important role for the full RAF kinase activity. And similarly, you see here that if you mutate V45E or D153A, these are actually results in almost 50 to to 60% reduction in the kinase activity. Again, these mutations don't perturb Ras-RBD interaction, so you're only perturbing Ras-CRD interaction. So it does look like that the Ras-CRD interaction is also important for Ras-mediated RAF activation, at least the full activation. Another thing which we noticed that in Ras superfamily, there is a Ras subfamily, and in the Ras subfamily, there are many proteins, which is again listed in this sequence alignment here, starting from K-Ras, M-Ras, RAP, RIN, RAT, RAL. Many of them have very similar switch one region, and many of them, if you interchange between these isoforms, they actually bind to the RBD. But what has been seen in many cases that many of these Ras subfamily members don't activate RAF. So why does that happen? And it does some of our structural work provides any clue about it. So one of the things what we did is to try to see what is the sequence conservation for the CRD Ras interaction interface. So you're looking at switch one region here, and you can see that majority of the residues, you can see, shaded black here, are conserved across all Ras subfamily members. But when you move towards this part, which is the interswitch region, especially these two parts, QVV, this is 43, 44, and 45. Here, the conservation becomes significantly poor in case of non-Ras family members. So you can see the K-Ras, H-Ras, N-Ras is QVV in all three Ras isoforms. But as you change to other, you can at least have one or two substitutions in these particular 3 amino acids. And the structure of M-Ras is known. And when we try to superpose M-Ras on top of K-Ras in our structure, you can see that this particular valine, which is V45 in case of K-Ras, is replaced by a glutamate 55 in case of M-Ras. And that may explain that, that even though it forms a complex with RBD, but when you have CRD, probably CRD is not interacting to M-Ras exactly the same way as Ras would interact. So just to understand this, we actually made a similar mutation in case of Ras and replaced some of these QVV in case of K-Ras by HTE. And as you can see that when we make that mutation, the level of phosphor goes significantly down in this case. So this does, some of these studies does indicate that interswitch region interaction with CRD might be responsible to some extent in the loss of Ras activation of other Ras subfamily members. We also went and looked at different mutants of Ras and how they interact to RBD CRD. We in fact went ahead and solved a structure of one each. One of them from G12, G12V, glycine 13, glycine 13D, and Q61R. And considering all three are oncogenic, as expected, they all bind to RBD CRD with slightly lower affinity than wild type, but very similar, not significantly lower. And as you can see in these cases, so here you see Q61R actually points towards an empty place on the Ras RBD interface, where it doesn't really perturb anything. So the interaction remains same. And this is G12V here in this purple, and then these mutations only change the position of Y32 because actually these two side chains clash with the wild type Y32 conformation and has some local effect on Y32 main chain. But other than that, these mutations have absolutely no effect on Ras RBD CRD interaction. And that basically explains how these mutations could still allow Ras-Raf interaction and remain oncogenic. And just for those of you who are not familiar with this, the GAP proteins bind at this site, and the arginine actually finger points towards the gamma phosphate from this side. And when you have this G12V or G13D, the arginine finger cannot position itself into the right conformation, and that's why it cannot hydrolyze GTP to GDP. But these, so in that case, in case of GAP, this becomes an issue, but in case of effector, this is not an issue. This is a local conformational change, but it still has no problem binding to RAF. We also wanted to, just last two slides, in this case, we wanted to check how does this whole complex could potentially stay on the membrane? As you know that Ras is a membrane, peripheral membrane binding protein. CRD is a membrane anchoring domain. So this actually gives us two anchors to place it. And based on previous results on how CRD interacts to the membrane, and we know that Ras has been shown to bind to membrane in many possible conformation. And so what we did that we took every possible conformation of Ras which has been proposed to bind to membrane, and then we let CRD decide which one will actually also allow CRD to interact with the membrane. And when we explored all possibility, this was the only confirmation of K-Ras membrane interaction by alpha 5 alpha 4 interface, which actually allows CRD to still interact with the membrane. And in this way, we believe that in this particular conformation, HBr is anchored in the membrane. CRD is at the same time anchored via these hydrophobic residues into the membrane bilayer, these arginine and lysines via the head groups, and interacting with the Ras via interswitch regions, and RBD is placed here. And again, these are previous residues identified from the previous NMR work, which have been shown to located on two hydrophobic loops of CRD. And this is the basic patch which potentially interacts with the membrane head groups. So just to understand how our work actually fits into the cryo-EM work, which was done by Michael X group, and actually, combining these two results together actually provides additional insights. So what you're looking at here is the structure reported by them. This is a MEK kinase domain, and this is BRAF, and this is 1433. As I said, that they were able to place the CRD in the middle. They had some partial density for RBD, but it was probably a little weak to place it. So what we did, we just placed the CRD on top of what was present in data structure, and that allowed us to see where the RBD could go in that complex. And then once we place the RBD, then we wanted to see how would the K-Ras would fit into that. And then as you see that when you place the both RBD, CRD and K-Ras, you can see that there is not significant a structural overlap between RBD, CRD, K-Ras complex and this auto-inhibited complex. So it looks like that when K-Ras comes in contact with RBD, it could also interact potentially with CRD, and then try to take CRD away from this autoinhibitory complex to convert this into an active state, which I've tried to show you in this last slide as a schematic. So this is what you see. Actually, we saw in the structure where you have this is the autoinhibited complex of RAF, where you have kinase domain, this RBD CRD, and these are the two phosphorylation sites, one on the C terminal end and one on the middle of CR2. And this is actually what RAF potentially looks like in the auto-inhibited conformation. When K-Ras comes close to it, we believe that the first interaction point is still RBD because it is fully exposed. And then the second contact point happens between Ras and CRD. And when that happens, it basically pulls it out of it. And then at the same time, CRD now is interacting to the membrane as well as to K-Ras. And as we suggested, this is really not a 2-bead on a string. This is a more like an extended structure of RBD CRD. And as it has been shown that it exposes the CR2 region, and which actually has shown to get dephosphorylated by a SHOC2 protein and releases this from the molecular handcuffing, which you see in the auto-inhibited state. And once this the molecular handcuffing is removed, as it has been shown that CRAF potentially forms a heterodimer with BRAF, and forms BRAF comes with MEK, and that's how it forms an active RAF dimer. So some of these work, our crystallographic work, along with the EM work which has been done together, does provide some insights on to how Ras interacts to RAF, and possibly how Ras allows it to overcome the molecular handcuffing and allows it to convert from inactive to active state. So that's all I had. Most of the work on the NF1 side was done by postdoc Upeng Yan, and on the RAF side was done by Tim Tran. The SPR work was done in collaboration with Andy Stephen here at some of the molecular modeling work was done by Chris Neal as Lawrence Livermore. Both project was NF1, spread one, as well as RAF was done in collaboration with Frank's lab at UCSF. At Ras Initiative, we have extensive protein expression lab, which provides us with the proteins we need, and then the leadership, Dwight and Frank. And as I said, I really like all the support which I get from SBGrid to allows me to focus more on the science, not so much on the updating and compiling different software. So thanks to SBGrid. And I also, I'm very thankful to Argonne for giving us generous beam time for these structural works. So thank you, and I'm happy to take any questions you may have.
Thank you. That was a couple incredibly detailed stories and a lot of work. And also I'm glad to hear that you have to worry about compilers less and you can focus on keeping your head straight with all of these problems.
Yes.
And so if folks have questions, feel free to send them to me in the chat or raise your hand and unmute yourself. I've got a couple that have accumulated as we've been going on. So I guess one of them for the K-Ras spread one structure, where you were talking about both of the extended structure, or the extended regions being necessary for binding. Is that because the N-terminal extension is stabilizing the C-terminal extension, or vice versa, or are they both actually involved in the binding interface?
Yeah, so I think that's a good question. I think we believe that, first thing, I think if you look at the interaction directly, it actually does involve both of them, but it looks like that the N part is a bit more important than the C part. But it does look like that both of them also form an intertwining. So if you carefully look at these, one helices goes on top of each other. So it does look like that they also somehow involved in a globular folding as well. So from that perspective, I think both of them needs to be together. But it does look like that extra domain on the N side plays a more major role than the C-terminal side.
So as frequently is the case in biology, when you have an either-or question, the answer is a mix of both.
Yes.
Thank you. Could you comment on the stability of the K-Ras and the hypervariable glycine-rich region and the chaperone protein?
Yeah, so I think that once you have that complex, yeah, so I think this is the structure. These are actually quite stable. So, you know, if you take any protein, and especially if you put this acyl chain, it has probably a 15 carbon long acyl chain, which is shown in yellow, which is you know, very hydrophobic. And so once it actually goes inside the protein, you don't really see anything. It actually completely shielded from the aqueous environment. So once you assemble this complex, they are very stable.
Thank you. So we have a question from the audience. So why is the energetic contribution of the CRD relatively small given the substantial interface it has?
That's a very good question. And I think, you know, considering when we looked at the, you know, Ras-CRD interface, when we saw 8 hydrogen bond, we did expect quite a significant. We believe that that RBD Ras interaction is just so small, just so strong, that some of these effects are very, very, you know, even if you are trying to disrupt it, it's not that significant. And as you have to notice that most of our mutations in this side, which we made, except maybe one, all of them were simple alanine mutation. We didn't really do a lot of charge reversal in this case, in at least on the CRD side, except in one case we did V45E. So we believe that also didn't really show the impact. But I believe that still, even the CRD plays a supporting role. The major interaction contribution comes still from RBD.
Thank you. And I just want to be respectful of your time. We've hit one, or do you have to hop off to something else?
I can take 1 or 2 questions still.
Okay, I will. So does NRAS have similar oncogenic roles as the HK and NRASs?
I'm not aware of it. I haven't seen, so I would say no, but I probably have to look into the literature. I'm not fully sure about it.
One last one, and could you comment on the potential interaction of KSR with K-Ras?
Yeah, so that's something we haven't done, and I'm sure Mark Therian's group have done a lot of work on KSR RAS interaction, and there is KSR as well as N-terminal, yeah, some of those interactions. We don't really have done any work on KSR, so I don't really have anything more to add to that.
There's always more questions than there are answers, it seems. And I guess I apologize. This really will be the last one. But is there any indication that there's post-translational modifications of CRD, and if those have any, would those be feasible, and would those be of potential biological impact?
Sorry, I missed the first part of the question. Can you?
For the CRD domain, are there any indications of potential post-translational modifications as far as something that's known or feasible?
Not, we didn't really. We haven't seen anything in the, at least we expressed many of them are in insect and mammalian. We haven't seen any post-translational modification in the CRD, at least in our part. And I think one thing which I just want to add, that there was a lot of some structural, some not a structural biochemical data that CRD could potentially interact to the farnesyl group of Ras. We did do a lot of studies both biochemically, and we couldn't really identify any interaction between CRD and the farnesyl group. So that's something which was reported in the literature 20 years ago, but we didn't really see any interaction there.
Thank you. And that's lots of incredibly interesting moving parts going on here. Thank you again for taking the time to present this today. This was really interesting stuff.
Oh, thank you for the opportunity. Happy to share our work. Thank you. Have a good rest of the day.
Yep, likewise.
Yeah, bye-bye.
Bye.